We live in a vibrant world, but have you ever stopped to ask what color actually is? We are about to embark on the fascinating journey from light waves to color perception. The first thing to understand is that color isn't inherent to objects themselves. It is actually a perception created by the brain based on how it interprets reflected light wavelengths. When you look at a red apple, it appears red only because of selective reflection. The apple reflects red wavelengths while absorbing all others. To understand vision, we must first understand the physics of light. Light travels in waves, and it is the wavelength that determines the color we perceive in the visible spectrum. This visible light is just a tiny slice of the much larger electromagnetic spectrum, which includes radio waves and X-rays. Within the visible range, the length of the wave dictates the hue. For instance, red light has the longest wavelength, measuring approximately 700 nanometers. If we move to the opposite end of the visible spectrum, we find violet. Violet light has the shortest wavelength, measuring approximately 400 nanometers. Every color we see exists between these two measurements. But what about white light? White light is actually the complete visible spectrum combined together. We can see this when we use prisms, which separate white light into individual rainbow colors. So if white is the presence of all light, what is black? Black is simply the absence of light. It occurs when objects absorb all wavelengths without any reflection. In this case, no light reaches our eyes, creating the perception of darkness. This interaction of reflection and absorption is how we dress ourselves daily. A blue shirt looks blue because it reflects blue wavelengths whilst absorbing red and green light. Conversely, a white shirt reflects every wavelength across the entire visible spectrum. Now that we understand the light, let's look at the machine that captures it. The anatomy of the eye. The journey begins at the cornea, the transparent protective outer layer. Light enters through the cornea, which begins the process of focusing incoming light. Immediately after, the light passes through the pupil. This is the adjustable opening that controls exactly how much light enters the eye, widening in the dark and shrinking in brightness. Once through the pupil, the light meets the lens. The lens is responsible for fine-tuning the focus. It projects the image onto the back of the eye, much like a projector in a cinema. This destination is the retina. Located at the rear of the eyeball, the retina receives this focused light and performs a miraculous translation. It converts light energy into electrical signals that the brain can interpret. The retina is not just a screen. It is lined with millions of light-sensitive cells called photoreceptors. These cells detect and respond to incoming light energy. There are two main types of photoreceptors that serve very different functions: rods and cones. Rods are specialized for low-light vision. They are sensitive to dim light and are excellent at detecting motion, but they only provide black and white vision. When the light for bright, the cones take over. Cones are sensitive to bright light and are responsible for our color detection. They also provide our fine detail perception. Humans typically have three types of cones, each tuned to a specific part of the spectrum. First, we have S cones, which detect blue by responding to short wavelengths. Next are M cones, which detect green by responding to medium wavelengths. The third type is the L cone. These detect red by responding to long wavelengths in the spectrum. You might wonder, if we only have cones for red, green, and blue, how do we see millions of colors? The answer lies in combination. Every color we perceive results from combinations of these three cone signals working together. It is a biological version of RGB technology. It is important to distinguish how eyes mix color compared to how we mix paint. With light, we use additive color mixing. Here, red, green, and blue wavelengths combine to create white light. However, with physical pigments like paint, we use subtractive color mixing. In this method, pigments like cyan, magenta, and yellow absorb wavelengths to create black. Once the rods and cones have captured the light, the signal must be processed before leaving the eye. Photoreceptors stimulate the intermediate bipolar cell layer. From there, the information flows forward through the retinal neural network to the ganglion cells. The axons of these ganglion cells bundle together, creating the cable that connects the eye to the brain, the optic nerve. The optic nerve carries these electrical signals from the eye to the brain for processing. However, there is a design quirk in the human eye. At the exact point where the nerve exits the eye, there are no photoreceptors. This creates a blind spot. We usually don't notice it because our brain automatically fills in the missing information based on the surrounding imagery. The signals travel from the optic nerve to the visual cortex, specifically reaching the occipital lobe at the back of the brain. Here, the brain acts as a decoder. It interprets the ratios of cone activity. For example, when both red, L cones, and green, M cones, fire strongly at the same time, the brain interprets this combination as the color yellow. This is how different combinations create our full color perception experience. The story gets more complex with the opponent process theory. After the cones detect light, ganglion cells process color information in opposing pairs. These pairs are red versus green and blue versus yellow. These create competing neural signals. This theory explains why we never see a reddish-green. We cannot see it because these signals oppose each other neurally. We can demonstrate this opponent processing with afterimages. If you stare intensely at a bright red image for 30 seconds without blinking, you fatigue your red channels. When you then look at a white surface, the opponent green response rebounds strongly. As a result, you perceive a green square phantom where the red one used to be, as your visual system recalibrates and recovers. Not everyone experiences color this way. Colorblindness is a genetic inability to distinguish colors. This vision deficiency results from missing or malfunctioning cone types. The most common form worldwide is the difficulty distinguishing red and green. This usually happens because the specific cones for those wavelengths are either damaged or entirely absent. Interestingly, men are much more affected by color blindness than women. The reason lies in our DNA. The genes for red and green color pigments are located on the X chromosome. Males have XY chromosomes, meaning they have only one X. If that one gene is faulty, they will be colorblind. Females have XX chromosomes, meaning they need two defective genes to show the condition. Otherwise, they are just carriers. Human vision is remarkable, but the animal kingdom sees the world very differently. Dogs, for example, have dichromatic vision, allowing them to distinguish only blue and yellow wavelengths. They see a limited world compared to us. On the other hand, many birds and insects can perceive ultraviolet light, a part of the spectrum completely invisible to humans. But the true champion is the mantis shrimp. While we have 3 cone types, the mantis shrimp possesses 12 to 16 types of color receptors. Finally, we must realize that our surroundings dictate what we see. Factors like lighting conditions play a huge role. Sunlight versus fluorescent light dramatically changes how we perceive color. Furthermore, simultaneous contrast means that neighboring colors influence our perception of each other. Individual perception differences even lead to viral debates, like the famous blue-black or white-gold dress, which showed how brain interpretation varies from person to person. To summarize our journey, it begins with physics, as wavelengths of light carry information. The eye's anatomy, cornea, pupil, lens, and retina work together to capture and focus this light. 3 types of cones detect the RGB wavelengths. Finally, the visual cortex processes these cone ratios, creating the rich, colorful experience of the world we enjoy every day. Thank you for listening.